Okay, let's go into chapter 10, meiosis and sexual reproduction. Those are not tadpoles. Okay, I want you to take a look at that illustration, though, for just a moment. I think that's just a miracle. Look at that. Obviously, that's an egg, and those are sperm. The egg is the largest cell in the body, and the sperm is the smallest cell in the body. Size differential. We'll talk more when we get into spermatogenesis and oogenesis about why the sperm is actually so small and the egg is large. Okay? Meiosis is a very specialized cell division that results in the formation or the production of sex cells, which are gametes. Meiosis is a very specialized cell division that results in gametes, which we call sperm and egg in higher animals. Okay? Gametes. Your normal body cells are diploid, and last week we called those somatic cells, right? They are diploid. So let's just take a look at this couple. Okay, who's that? Homer and Marge, yes. So their somatic cells have the diploid number of chromosomes, which is 46. Well, we know that Marge is producing eggs and Homer is producing sperm, right? Well, if you didn't have any special type of cell division, that means that your eggs would have 46 and your sperm would have 46, right? Well, when those two nuclei unite, if that was the number in the egg and the sperm, then you would not have a normal human, okay? Right. That would be doubling the chromosome number with every generation, okay? So you can't do that. You cannot double the chromosome number. We learned last week that every species has a certain number of chromosomes, right? Well, 92 is not a normal human. Yeah. So you have to have some type of division that's going to take the diploid number and reduce that number so that the gametes have half that number. So when your gametes fuse, you restore that diploid number again. And that type of division is called meiosis. Okay? We sometimes call meiosis reduction division because that's what's happening. In meiosis, a normal diploid cell is taken through divisions, and the result of that are haploid gametes. Okay, it's a very specialized cell division, and it only occurs in very special tissues. Okay, those tissues for higher animals are found in the ovary and the testes. Okay, it's going to reduce our chromosome number from diploid to haploid, which we said last week that diploid was 2 of each kind, and haploid is 1 of each kind. So we're going from 2N to N, from 2 of each kind to 1 of each kind. And diploid in humans is 46. What's haploid in humans? 23. That's exactly right. So we're going from 46 to 23. This is in meiosis. And then again, the fusion of those two gametes would give you 2N again, right? Every gamete would be N, and then you get 2 nuclei fusing together, and you would have 2N. Okay, so now the egg would have 23, and the sperm would have 23, and you would have a normal number again. So you have to reduce that chromosome number by half, 1 of each kind that goes into the gametes. Okay? Okay, that's what sexual reproduction is. When we talk about things reproducing sexually, that means that they're producing these specialized cells that are haploid, that fuse to form a new individual. So it's the forming of a new individual by the union of sex cells. Or in higher animals, we call those gametes. Okay? Sexual reproduction was an evolutionary advancement There are lots of benefits to reproducing sexually. So let's talk for just a moment about asexual versus sexual reproduction. If you guys remember, we said that for organisms that reproduce asexually, they produce offspring that are genetically identical to themselves. It's like a clone, okay? So that you would have very little variation in the population. If I reproduce asexually, all of my offspring are genetically identical to me, okay? So this type of reproduction, asexual reproduction, is okay as long as the environment is stable. Okay, so if the environment is stable, then asexual reproduction has an advantage. But for environments that are not stable, for instance, there are some organisms that can reproduce both sexually and asexually. If you take 104 next semester or whenever, you'll learn about some of the lower animals that can actually reproduce asexually. Well, asexual is fine as long as conditions are stable. But when conditions get uncertain and survival is questionable, then those organisms oftentimes will resort to sexual reproduction to get some genetic variation so somebody might survive. Okay? So when things are not stable, sexual reproduction has an advantage, okay? So what does sexual reproduction do? It gives you gene variety, and with gene variety, you get disease resistance, so somebody might survive, okay? Increased disease resistance. So what organisms reproduce sexually? Plants. Plants actually reproduce sexually. Fungi reproduces sexually, and animals, higher animals, reproduce sexually. So all three of those have sexual reproduction in those kingdoms. So where does it occur? Well, in higher animals, in the female, there are ovaries that produce eggs. There are testes that produce sperm. These are the germ tissues. These are the only tissues in those organisms that will undergo meiosis. This is the only place where reduction division will take place. Okay? Collectively, we would call the egg and the sperm gametes. In plants, the female portion of the plant is called a pistil or a carpel in some books. The pistil is this portion right here. There's the ovary. There's the ovule right there. The male portion is called a stamen. That's this structure right here with the filament, and the anther is the yellow structure right there. Collectively, the egg and the sperm are called gametes. They're haploid. And the ovule and pollen are spores that produce the sperm and the egg. Any questions? Okay, so let's make sure we have our reproductive vocabulary down so that when we say these words, everybody understands what we're talking about. We're going to talk about meiosis specifically in higher animals. We're going to use humans as an example primarily, okay? So, sperm is a male sex cell. The egg is the female sex cell. Collectively, we call those gametes, and by definition, a gamete is haploid. And we said last week that gametes are produced by germ tissue. And tonight we learned that that germ tissue is in the ovary and the testes, okay? Somatic cells are body cells, and by definition, they're diploid. Union of the nuclei of those two gametes is called fertilization, and the resulting cell that's now diploid is called a zygote. Everybody got it? Vocabulary down? I love that illustration. Look at how many sperm there are to that one egg. Look at that. Is that not amazing? I mean, there are cajillion sperm produced, but only one egg a month. Why do y'all think that is? Any hypothesis about why one? Yes, sir. Do what? The female body couldn't handle more than, yeah, that's probably true. Huh? Did you just get it right the first time? I love that. I really have this theory that it's because sperm will never stop and ask directions. I'm just kidding. Okay. Okay, so last week we talked about homologous pairs. Does anybody remember what homologous pairs were? Do what? What's a homologous pair? They're similar. Yes, yes. These are those two that look alike, right? We said they looked alike. So let's talk about what that means to look alike, okay? We said humans had 23 pairs, right? Females have 23 homologous pairs. Males have 22 homologous pairs and an X and a Y because the X and the Y do not look alike, okay? Okay, so these are pairs of similar chromosomes. So when I say that they're similar, that means that they're the same length. Okay, so obviously looking at that karyotype up there, there are some that are longer than the others. Okay? And you know where the centromere is? We talked about the centromere last week. The top portion is the P portion of the chromosome, and the bottom is the Q. So on some of them, the centromere is in the middle, so P and Q are equal. On some of these pairs of chromosomes, the centromere may be closer to the top. So the P would be really short, and the bottom would be long, okay? So they're about the same length. They have the same centromere location. And look at that illustration there. Those chromosomes have been stained. Look at the staining pattern for those chromosomes. You can really tell which ones are homologous when they're stained. Why? They have the same stripes. Look at they're striped, right? They look exactly alike. They have the same banding pattern when they're stained. So you can really tell which ones are homologous when they're stained, okay? So let's talk about what that really means to be homologous, okay? I'm going to go ahead. I'm going to say this. We're going to get it in your notes, and then I'll explain it, okay? Homologous chromosomes, those that are homologs, have the same genes for the same traits in the same positions. They have the same genes for the same traits in the same positions. Now, what in the world does that mean? Let's talk about that. That means that, let's take this illustration right here that says pair number 1. Let's say this is your pair number 1, okay? So let's talk about what exactly that is first. Remember that each one of these is a chromosome, so prior to replicating and condensing, that was a piece of chromatin, right? That replicated, made a copy of itself, and then condensed, so now it's a chromosome. So whatever is on this left side of that chromosome is exactly identical to what that right sister chromatid is, right? So these are identical to each other. You can see, I mean, they're the same color. They've color-coded them, okay? So here's your pair number 1. One of these came from the egg that formed you, and the other one came from the sperm that formed you, right? So you have 2 copies of the sperm's information and 2 copies of the egg's information for whatever genes are on that chromosome, right? Okay? So they have the same genes for the same traits in the same locations. 2 seconds. Okay, same genes for the same traits in the same locations. So, hypothetically, if the gene for eye color is right here, okay? If the gene for eye color is right there, then we know exactly where that gene is, okay? We know which base pair it starts on, how many base pairs are included in the gene, and which base pair it ends on. So the gene for eye color would be in that same place for every single Homo sapiens chromosome number 1, okay? So your dad's chromosome number 1, the one he gave you, and your mom's chromosome number 1, the one she gave you, are going to have the gene for eye color in the exact same place. Okay? So the same genes for the same traits in the same place. Okay? Now chromosome pair number 2 would not have that gene. Every pair of chromosomes has a unique set of genes. If we think about all 46 chromosomes as a cookbook, you could think about the pair as a chapter in that cookbook. There are certain genes on pair number 1 that aren't anywhere else. There are another subset of genes on pair number 2 that aren't anywhere else. So every pair has its own specific set of genes on it. Okay? These are not identical. I've never said homologs are identical. I've said they're similar. Okay? Because if this is the gene for eye color, your mother may have given you a gene that codes for blue eyes for that gene. Your dad may have given you a form of the gene that codes for brown eyes. So the information there is not the same, but the gene is coding for the same trait. Does that make sense? So that's what homologs are. The variants of genes are called alleles. So mom's information would be an allele for blue eyes. Dad's information would be an allele for brown eyes. So the gene is eye color. The form that gene could take, blue eyes, brown eyes, etc., is called an allele. Okay? If we're talking about plants, the gene could be for height in a plant. The alleles might be tall and short. The gene might be for flower color. The alleles might be purple and white. So the allele is the form that gene would take. Okay? These are not identical. One came from your mom, one came from your dad. And we would say that they're homologous to each other or they are homologs. Okay? They're homologous or they're homologs. Diploid cells contain 2. Okay, let's talk about what diploid actually means now. We said 2 of each kind, right? Meaning that one of them came from your mom and one came from your dad. Diploid is actually when you have those 2 alleles in the cell together. So when you have mom's information for that gene, that allele, and dad's information for that gene in the same cell, that's diploid. Okay? So it doesn't matter, it doesn't have anything to do with how many copies there are. Back when these were chromatin, you still had mom's allele and dad's allele, right? So even when they were chromatin, that's still diploid. When these separate from each other, as in gametes, then that's haploid. When you only have one allele in the cell for that particular trait. Okay? Haploid then would have 1 from each pair. Everybody still with me? Okay. Here are karyotypes. When you do a karyotype, if you have a genetic issue in your family, the first thing that the geneticist is going to do is a karyotype. He or she is going to want to see what your chromosomes look like to make sure you have the right number and that they're structurally sound. Okay? So when you do a karyotype, keep in mind the only time we can see chromosomes is close to division, right? After they condense. So the geneticist would have to find a cell in division, or close to division, and stain the chromosomes. And it would look something like that, okay? And then what they do is they literally, used to, before computers, they literally would cut them out like paper dolls and find the ones that were homologous and put them side by side. And when they put them on the karyotype, they always start with the largest pair, and they end with the smallest pair, reserving the 23rd spot for the sex chromosomes, always. Okay? So this is obviously a female. That is a male. And don't let it confuse you that some of these are bent over. They do bend sometimes. Those are still homologous. And you can see here that the 23rd pair on that particular karyotype, the Y is not anything like the X. So those couldn't be homologous. Okay? Just a little note as we go into the rest of chapter 10. The X chromosome, these are called the sex chromosomes, okay? But the X chromosome actually contains genes that are non-gender related, okay? The X chromosome contains genes that deal with being a human being. So every viable, which means living, every viable human being has to have an X chromosome, okay? You can live without a Y, obviously, because girls have two X's, right? You can live without a Y, but you can't live without an X, okay? As a matter of fact, toward the end of this chapter, we're going to talk about some conditions where people get the wrong numbers of chromosomes. And sometimes individuals get too many or too few sex chromosomes, okay? The presence of the Y is always going to make the person a male, okay, unless there's some type of receptive issue on cells, okay, where the hormone's not actually received. So the presence of a Y. So if you have an individual that has two X's and a Y, have an individual with 3 sex chromosomes, that individual is going to be a male. Even though they got 2 X's, the Y is going to make that person a male, okay? So you can live without a Y, but you can't live without an X. Okay, let's pause for just a moment. Okay, I'm not sure what happened to the microphone. May have to fill that in. Okay. Okay, so we're going to take the diploid cell that just came out of interphase in an ovary or a testes. We're going to take it through 2 divisions. Meiosis 1 has a prophase, metaphase, anaphase, and telophase. That cell would divide into 2. And those 2 cells would undergo meiosis 2, which has a prophase, metaphase, anaphase, and telophase. Okay, let's step back for just a moment and talk in general. Go back to what we talked about in mitosis. What were some of the general activities that were happening in a prophase? What was happening in a prophase? Do what now? Nuclear envelope's disintegrating, nucleolus is disintegrating, spindle fibers are forming. Yes, so those are some general activities that always happen in prophase. Okay, so in both of those prophases, you would expect that to be happening. So what's happening in metaphase? It's lining up in the middle, right? Something's lining up in the middle. okay, in metaphase. So what's happening in anaphase? Something's coming apart, right? What's happening in telophase? We're going to have two cells forming, spindles disappearing, nuclear envelopes coming back, etc. Okay? So those same general activities. You can do your same dance for all of these, okay? So now we've just got to talk about what is it happening to? What's lining up in the middle? What's being separated? Okay? So remember, we started this with a genetically normal diploid cell, replicated condensed chromosomes, right? Right? Okay. The purpose of meiosis 1, what is happening in meiosis 1? The purpose of meiosis 1 is to separate the homologous pair. Okay, so we're going to separate the homologous pair. Members of homologous pairs separate from each other. Separate the homologous pair. Okay? What does that mean? Anybody want to add? Separate the homologous pair. If I have a homologous pair of chromosomes, they look alike, right? There's a red chromosome made out of beads. There's a yellow chromosome made out of beads. They both look alike, except they're not identical, right? If I separate this homologous pair, that means that this one is going to go into a cell, and this one is going to go into another cell, right? And that would be happening, if we were talking about a human cell, that would be happening with all 23 pairs. One of each pair would go into a cell, and the other one from the pairs would go into another cell. That's separating the homologous pairs. Okay? So how many would this cell on my right have? 23. How many would this one have? 23. And each one of those would now be haploid because I separated the pair. Separation of the pair equals haploid cells. Okay? What's the purpose of meiosis 1? Separate the homologous pairs. What's the purpose of meiosis 1? Separate homologous pairs. What's the purpose of meiosis 1? When do the homologous pairs separate? What's the purpose of meiosis 1? What is the purpose of meiosis 1? When do pairs separate? What happens in meiosis 1? Okay, so you just told me in one of my cells, if we're talking about human, in one of my cells I got 23 chromosomes, one from each pair. And in the other cell I got 23 chromosomes, one from each pair, right? Those two cells are now going to undergo meiosis 2. Okay? So what happens in meiosis 2? What is the purpose of meiosis 2? Now I've got 23 chromosomes. The purpose of meiosis 2 is to separate the sister chromatids. To separate the sister chromatids. What's the purpose of meiosis 2? Separate the sister chromatids. What's the purpose of meiosis 2? What's the purpose of meiosis 2?
Separate the sister chromatids.
What's the purpose of meiosis 2? Okay, so I've got 23 chromosomes over here in this cell, right? What am I doing? I'm separating the sister chromatids, okay? So, 23 pairs of homologous chromosomes. Meiosis 1 separates all the pairs. Meiosis 2 separates the sister chromatids, right? So I end up with 4 cells that have single strands of each type of chromosome, right? You still with me? Okay, very good. Here it is, guys. This is really not hard at all. Look at this. Look at my beginning cell. That's G1. I don't know if you realize it now or not, but G1 is the only time that you see single strands of chromatin, right? G1 is the only time that you would see a diploid cell with single strands of chromatin. Okay, G1, we had to go through an interphase, right? What happens in interphase? G1S and G2. So we replicated and condensed, right? So now we have chromosomes. This is my cell that's going to undergo meiosis 1. What's going to happen in meiosis 1? The homologous pairs separate. There's my pair. The blue one is over here on the left now. The red one is over here on the right. My small pair right there, the red one, is now over here on the left. And my small blue one is now over here on the right. Meiosis 1 separated my homologous pairs. So there's only one of the long ones and one of the short ones. If these were human cells, there would be one number 1, one number 2, one number 3, one number 4, etc. Okay? This cell is diploid because now, with regard to the genes that were on that big chromosome, the only representative in this cell are the genes that came from the maternal contribution, right? My genes from the paternal are over there. Okay? Diploid haploid has nothing to do with the number of copies. It has to do with the alleles, the genes separating. Okay? Meiosis 2 did what? That chromosome tore apart. There's my long red one there, my long red one there. This small blue chromosome tore apart. There's one blue, and there's one blue. So I now have 4 cells that have one single copy of each kind of chromosome. Okay, let's get it in your notes. We start with a diploid cell. We're going to go from diploid to haploid. We're going to do it in 2 divisions, meiosis 1 and meiosis 2. We are going to produce 4 haploid cells. They're all going to be genetically different. Each cell is going to get 1 from each homologous pair. And again, remember, always remember, this only occurs in the tissues of the ovary and the testes. These are the only types of cells that undergo this specialized cell division. It's the only place gametes are found, are produced. Okay, everybody with me? Okay, so let's go through it step by step because there's some other things that happen too. Okay, it's not quite exactly. I mean, that happens plus some more things. Okay, so let's go through it step by step. We're going to start, this is our cell in G1, right? Single strands of chromatin. Again, the book uses this diploid illustration with 4. We'll use this for the next several chapters. Our diploid number here is 4. Okay, if that were a human cell, there'd be 46 of those up there, right? 23 red ones and 23 blue ones, right? Okay? That's diploid. So what is, if that's 2 long ones and 2 short ones, what's haploid? 1 long and 1 short, right? It's going to be half of that, 1 from each pair. Well, something really, we call this reduction division. I think it's kind of ironic that before we even go into reduction division, we replicate everything. So instead of reducing, we just doubled, right? So we had to go through G1, S, G2. So we just doubled everything. So now how many haploid cells can I make? Every haploid cell has 1 long and 1 short. I can make 4 now, yes, okay? I can make 4 haploid cells now, okay? Okay, now we're getting ready to go into what? Meiosis 1. prophase 1, okay? The very first thing that happens, prophase 1, guys, this is hugely important. Star, underline, tattoo this on your palm, okay? This is extremely important. This is what makes meiosis, reduction, division, okay? The pairs find each other. Prophase 1, the homologous pairs come together and they synapse. Synapse means they come together, very close. They come together. The homologous pairs come together during synapses. They synapse. So what does that mean? Well, that means that the long ones are going to find each other, and the short ones are going to find each other. And if it were human, all 23 pairs would come together. And we really don't understand why, but in a male, the X and the Y come together by default. Okay? So the pairs come together. That's called synapsis. They come together as if they are long-lost lovers. They haven't seen each other in ages. And guys, they get so close together, their legs wrap around each other, and they form what's called a tetrad. Okay? This is a tetrad right there. So that's the long pair that are together and all wrapped up in each other, and that's the short pair. Okay? Tetra, anybody ever played Tetris? Everybody's played Tetris. What does tetra mean? 4. That's exactly right. That's why it's called a tetrad. Because normally chromosomes are not blue and red. So if these were not colors, then that would look like one entity that had 4 legs. Okay? So it means 4. That's exactly right. So when this tetrad forms, something sometimes happens. It doesn't have to happen. You never know when or where it's going to happen, but sometimes the legs break off. Okay? I mean, these things are so wrapped up in each other that they break the legs and they attach to the wrong chromatid. It's called crossing over. Okay? So crossing over can occur. Okay, let's talk about that for just a moment, because this is really important. Crossing over is sometimes hard to understand the significance of it. So let's take a look at this pair number 1 as it synapses and forms this tetrad. Okay, crossing over is the exchange of genetic material between non-sister chromatids during meiosis 1. So we have synapses forming this tetrad. Exchange of genetic material. Okay, we've numbered the chromatids for convenience, so we can talk about them, okay? So this is the maternal number 1, and that's the paternal. So let's get this in your mind now, that this is your pair number 1, and now you're making gametes, okay? So this was the number 1 that your mom gave you, and this was the number 1 that your dad gave you. So I'm going to number these chromatids. Those are sister chromatids. Those are sister chromatids, right? That's 1 and 2, and that's 3 and 4. Okay? So they come together, they synapse, and they break. So what has happened in this illustration is chromatid number 1 broke, but it reattached to the wrong chromatid. It actually reattached to chromatid number 3. Okay? And chromatid number 3 broke and reattached to chromatid number 1. Those are non-sister chromatids. Okay? So look at what you get here. If that happens, you get chromatid number 1 that has some of the blue in it. Okay? And you get chromatid number 3 that has some of the red in it, right? So let's think about what this actually means. Okay, you saw a while ago in the example of meiosis that each one of these is going to go into a different gamete. Chromatid number 1 is going to go into a sperm. Chromatid number 2 is going to go into another sperm. Chromatid number 3 is going to go into a sperm, and 4 will go into another sperm. So you're going to be producing 4 gametes. Okay? So each gamete is going to get one of those. Well, think about this for just a moment. let's say hypothetically that the gene for eye color, and this is very simplified, but the gene for eye color is up here in the top region. And the gene for hair color, or a gene for hair color, is down here. Okay, so let's think about what this means. If chromatid number 1 sperm, if the sperm that gets chromatid number 1 happens to get to the egg first. Now keep in mind, there's another individual producing a gamete also, okay? So this is just your contribution to your future offspring, right? So if the sperm that gets chromatid number 1 happens to make it to the egg first, that means that your contribution to your future offspring would be the allele for eye color that you got from your dad and the allele for hair color that you got from your mom. So it would be that offspring's grandfather's allele for eye color and that offspring's grandmother's allele for hair color, right? If the sperm that gets chromatid number 2 happens to make it to the egg first, okay, then your contribution to your future offspring would be the alleles that you got from your mom for both eye and hair color. If sperm with chromatid number 3 in it gets to the egg first, then your contribution to your future offspring would be the allele for eye color that your mom gave you and the allele for hair color that your dad gave you. And if the sperm that gets chromatid number 4 makes it to the egg first, your contribution to your offspring would be both alleles from your father. Sexual reproduction is all about mixing up the genes as much as possible. Genetic variation. Okay? Okay, so where did we get to? We went through prophase. What's that? What's that? That is synapsis that formed a tetrad. That's right. Metaphase. After this occurs, then this tetrad, the spindle fibers are going to move the tetrads to the center of the cell. And the tetrad is going to separate so that you get the homologous pairs right next to each other in the middle of the cell. Because keep in mind, what did you tell me the purpose of meiosis 1 was? Separate the homologous pairs. Guys, if you're going to separate the pairs, you've got to have them next to each other in metaphase. Okay? Because remember, those spindle fibers are going to pull toward the centrioles. If you're going to separate that chromosome needing to go to that side and this red chromosome needing to go to that side, then you're going to have to put the pairs next to each other. Okay? So metaphase lines up the homologous pairs. Anaphase is going to pull the pairs apart. Telophase, you're going to get 2 cells, 1 from each pair in each of the chromosomes. Give you a moment to catch up. So what did you do at the end of meiosis 1? You separated the homologous pairs. Yes. So now you have 2 haploid cells. That cell's haploid because the genes, here's my long chromosome right there. You can't ever take into consideration that the crossing over occurred because it doesn't have to, and it might not. Okay, you don't know when it's going to happen, so you can't say, well, there's some red right there, so that's not haploid. That's haploid because that's chromosome, the long chromosome, and there's the other long chromosome, and they separated from each other. Crossing over doesn't have to take place. After telophase 1, you have a period called interkinesis where nothing happens genetically. So these 2 cells are exactly what go into meiosis 2. So I'm going to take these 2 cells straight into meiosis 2. And what's the purpose of meiosis 2? separate the sister chromatids. Yeah, so let's look at that. Prophase, spindle fibers forming. I'm going to attach to my chromosomes there. I've got my one from each homologous pair in those cells. Metaphase, my chromosomes line up right there. Anaphase, my sister chromatids pull apart, move toward the pole. Telophase, I have 4 cells that are produced with one chromatid from each pair. And then I have 4 haploid daughter cells. If you will just remember, meiosis 1 separates the pairs. Meiosis 2 separates the sister chromatids from each other. If you will remember that, you won't get as confused. Okay, so there it is again. Starting with a cell in G1, you have to replicate condensed chromosomes. Those go into meiosis 1, where your pairs separate. Each one of those goes into meiosis 2, where your sister chromatids separate. Let's watch this. We need to watch this. We need to process. Y'all are looking kind of scary.
Is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes.
Okay, do you understand what he's saying? When these things come together, what stage is this? Huh? Metaphase 1, right? Our pairs are lining up, right? When they come together, right here in the center of the cell, which one ends up on which side is arbitrary. It's by accident, okay? The way that they lined up in this example is the purple long one is on top right here, and the green short one is on top. It could have just as easily been that this purple short one would have been on top. keep in mind, you saw a while ago, ever how they line up in metaphase 1, those are going to go into secondary cells together, which will determine what the gametes are. Okay, so right now, the gametes are going to be those on top and these on bottom. Okay, so it could easily have been swapped, which would be an entirely different person, an entirely different gamete. Okay, you can mathematically calculate how many different combinations there are. The mathematical formula for that is 2 raised to the number of pairs of chromosomes. So for an example that only has 4 chromosomes as a diploid number, it would be 2 times 2. So there's only 4 different combinations, okay, which doesn't seem like a very big deal, okay, until you start thinking about humans. 2 to the 23rd is over 8 million different combinations. So you are who you are today just because these lined up arbitrarily like they did, okay? If one had swapped, you would be different. Okay? It's called independent assortment.
The chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber, and in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Sorry, guys. What am I doing? Who knows? Okay, right here. So, 3 ways that meiosis ensures genetic variability. Well, we have crossing over. We saw what that was, where homologous chromosomes come together in prophase 1 and synapse to form a tetrad, and sometimes crossing over takes place. Independent assortment, we just talked about. These homologs align at the equator in metaphase 1, and how they come together in the middle of the cell is arbitrary. So take a look at this example right here. This example actually has 3 pairs of chromosomes, so that the number of different combinations would actually be 2 raised to the power of 3. So it would be 2 times 2 times 2. So you've got 8 different combinations there. Notice in the first one, all the maternal chromosomes are on the right, the paternal on the left, and then they've mixed it up. So you have 8 different combinations there. Maternal or paternal homologs on either side. all different combinations. And then again, remember, possible number of combinations is 2 raised to the number of pairs. Remember that this is sexual reproduction, so it's going to take 2 gametes to form a new individual. So you've got to have 2 parents. And remember that both parents hopefully are genetically different, okay? And both of those gametes are going through this exact same process. So not only are your gametes crossing over and assorting independently, but your mate's gametes, chromosomes, are also doing that. So that when you get the gametes that fuse, then you have those parental genes combining. Guys, I don't want you to learn these numbers, but I just want to show you some statistics. Remember, sexual reproduction is all about mixing up the genes and making everybody different, right? If both gametes are undergoing this independent assortment, I'm not saying there's any crossing over, say there's no crossing over at all. Both gametes just going through independent assortment with 23 pairs of chromosomes gives you that many different possible different zygotes. Okay? One crossing over event, just one, from one of the parents. Okay? One crossing over event changes that number to that. So, are there ever going to be any 2 humans ever alike? Ever? No. Okay. Some comparisons between meiosis and mitosis. Take a look. You've got them side by side there. Remember, mitosis occurs in somatic cells. You start diploid and you end up with 2 diploid cells, right? And this would be your skin cells or your liver cells or muscle cells, okay? Meiosis occurs in germline tissue, and you get gametes, right? Okay, so meiosis is used for sexual reproduction. Mitosis is used for asexual reproduction and growth and repair. So 2 different purposes, 2 different divisions. 2 nuclear divisions in meiosis, 1 in mitosis. In meiosis, the chromosome pairs come together. And you can get that crossing over. In meiosis, the pairs come together. That's what makes it meiosis, because that doesn't happen in mitosis. The pairs don't care about each other in mitosis. They don't synapse. This is the difference right here, guys. Huge difference. Pairs together right here so that pairs can separate. Here, the pairs don't care about each other. The chromosomes are lining up. Homologs align, chromosomes align. You get 4 genetically different haploid gametes. They're different from each other. They're different from their parents. They're haploid. In mitosis, you get 2 genetically identical diploid cells. Okay, skip this slide. If you take 104, we spend a whole lot of time on that. Go down to the human life cycle. This slide just shows you where mitosis and meiosis occurs in your life cycle. Let's start with the production of gametes. It's a cycle, so it goes in a circle. We got to start somewhere, though. So let's start with the production of gametes. Your germline tissues in the ovaries and the testes undergo meiosis to produce haploid eggs and sperm. Those haploid eggs and sperm nuclei then unite, and you end up with something called a zygote. That's They unite, the nuclei unite, that's fertilization. You end up with a diploid zygote. And as we talked about earlier, that genetic information present in that zygote is the instructions for that new individual. This is a one-celled form of a new individual. Everything in that cell has already been determined. That individual's characteristics are determined in that one cell right there. So that is a single-celled stage of a new individual, of the next generation. Mitosis then occurs to this zygote so that that genetic information is copied into 2 cells and 4 cells and 8 cells and 16 cells. You end up with a multicellular embryo, a fetus, and then more mitosis. All growth is mitosis, okay? Growth is adding cells, so all growth is mitosis. So eventually this fetus and embryo will start to take on the characteristics of that individual as it was determined to be when the zygote was formed. All growth occurs as mitosis. So growth, development into adulthood, and then at adulthood, those germline cells start undergoing meiosis and producing gametes. So as a result of mitosis, each somatic cell in your body has the same number of chromosomes and the same genetic makeup that was determined when the zygote was formed. Any questions? Okay, meiosis occurs, go back, scream at me. You can scream at me. Whoa, slow down. We okay now? Everybody good? You good? Got it. Okay, meiosis is part of larger processes. In the male, it's called spermatogenesis, and the result is sperm. Spermatogenesis produces sperm, and it occurs in the testes. It starts at puberty. Meiosis is part of a larger process in females called oogenesis. Oogenesis is the production of eggs. And for females, it actually starts before birth and ends at menopause. So ladies, what does that mean? That means that you were born with every egg you'll ever have. It's like giving a little baby girl a carton of eggs and saying, take care of these. You might want to use 1 or 2 of them in about 25 years. So you have every egg that you will ever have. Okay? Okay, so let's talk about spermatogenesis just briefly and see how meiosis fits into it. It all begins with a stem cell called a spermatogonia. The testes contain stem cells called spermatogonia. These spermatogonia undergo division and produce the primary spermatocytes. Okay, so let's look at that. Spermatogonia make the primary spermatocytes. And it is that primary spermatocyte that's going to undergo meiosis, okay? So we've got something over here. The spermatogonia is not even in the illustration. We got a spermatogonia, that's our stem cell, that's going to divide and produce this cell right here. So we've gone through interphase. This is after interphase, okay? So we've got a primary spermatocyte. This has replicated condensed chromosomes. So we're ready to go into meiosis 1. What's the purpose of meiosis 1? Separate the homologous pairs. So in this secondary spermatocyte, I get half of the pairs, and in this secondary spermatocyte, I get the other half, right? So if this were human, I'd have 46 here, 23 and 23, okay? These primary spermatocytes undergo meiosis 1 to form those 2 secondary spermatocytes, okay? The two secondary spermatocytes undergo meiosis II, which, what's the purpose of that? Sister chromatids. So my 23 chromosomes right here are going to split apart so that I have 23 pieces of chromatin here and 23 here, right? So my secondary spermatocyte is going to undergo meiosis II to form spermatids that are haploid single copies, and they differentiate into sperm. So for every primary spermatocyte going into meiosis, I end up with 4 sperm. Look at the size, though, y'all. Look at the size differential there. We basically took the cytoplasm of that primary spermatocyte and divided it into 4, right? And then after differentiation, they still are smaller, very, very small, extremely small. These cells actually have very little cell content. They're not intended to survive. They don't have enough cell content to survive. They've got some mitochondria because they need the energy to propel the flagella, but these cells are not designed to survive past a few days. This is a mobilization unit for DNA. Its job is to get the DNA to the egg. Okay, they're not designed to survive. Okay, so that is spermatogenesis. All 4 become sperm. Let's look at oogenesis because it's different. Okay, same basic concept as far as division is concerned, but the fate of the secondary cells are different. Okay. So oogenesis, we've got the ovaries that contain stem cells called oogonia. The oogonia produce the primary oocytes. So here's my primary oocyte. It is the primary oocyte that's going to undergo meiosis. So the primary oocyte begins meiosis I, but only a few continue at the sexual maturity of the female. Okay? So we've got this primary oocyte. It's beginning meiosis I. At sexual maturity, it's going to continue. Okay? The primary oocyte is going to undergo meiosis I and form this secondary oocyte and something called a polar body. So let's talk about that. Primary oocyte undergoes meiosis I and forms a secondary oocyte and a polar body. Okay, so what happened here? Primary oocyte, you've got, if this was a human cell, you've got 46 replicated condensed chromosomes, okay? The nucleus is going to undergo meiosis I and cell division, and division, okay? So we're going to get 2 nuclei. There's one nucleus, there's the other nucleus. So we got our chromosome pairs separated into 2 nuclei, okay? Cytokinesis that accompanied it didn't go so well, okay? When the cells divided, instead of getting 2 equal size cells, one of the cells got most of the cytoplasm, and the other cell didn't get much cytoplasm at all. It didn't even get enough to survive. So you've got half of the chromosomes in here, 23, one from each pair. You've got half of the chromosomes in here, 23, one from each pair. But this cell didn't get any cytoplasm. So it can't survive. It disintegrates. It dies. It's gone. It's reabsorbed. Okay? So the only thing that you have that goes into meiosis II is this secondary oocyte. Okay? Notice how the egg is staying big. Okay? Secondary oocyte begins meiosis II, but it stops at metaphase II. The secondary oocyte begins meiosis II, but it stops and is ovulated. So that's when ovulation occurs. Okay? It's going to leave the ovary and it's going to enter the fallopian tubes. Now, what happens to this secondary oocyte, the fate of that cell depends on whether there's sperm present. Okay? If there is no sperm present, that cell, that secondary oocyte, disintegrates. Okay? And then menstruation would occur and everything would start over. Okay? If there's no sperm, the secondary oocyte disintegrates. If there is sperm present, okay, it is the penetration of that sperm through the egg. Now, this is not fertilization, okay? Fertilization is defined as the combination of the two nuclei, okay? It's the penetration of that sperm that once that sperm is there, the barrier set up, no other sperm can enter, that triggers this secondary oocyte to undergo meiosis II, because remember, we're not finished. We stopped at metaphase 2. So the presence of that sperm right there triggers that secondary oocyte to complete meiosis II. What's the purpose of meiosis II? Separate sister chromatids. So we're going to take this nucleus that has 23 chromosomes, if it's human, and we're going to tear those chromosomes apart and put one chromatid, one sister chromatid over here and one over here for all 23 of them, right? So nuclear division goes great. I end up with 2 nuclei. There's half of my chromosomes, 23 of them, one copy. There's the other one right there. Again, cytokinesis doesn't go so well. This cell doesn't get much cytoplasm, so there's my second polar body, and it disintegrates. So all I have left here now is 23 single strands, right? Which is exactly what I need. And then fertilization takes place. The 2 nuclei will combine. Isn't that a miracle? Any questions? Let's look at the spermatogenesis and oogenesis side by side. Sometimes that helps. Okay, spermatogonium, right there. Single strands, diploid, got to go through an interphase. Okay, if that were human, there'd be 46 of those. Interphase, production of the primary spermatocyte right here. We've got replicated condensed chromosomes. Meiosis I, separate the pairs. Meiosis II, separate the sister chromatids, differentiate into sperm. So that's spermatogenesis. Oogenesis, we start with the oogonium. We got to go through the interphase, replicate, condense. Okay, now we have diploid chromosomes. Meiosis I separates the pairs. There's half of my pairs. The other one goes into a polar body and disintegrates. Meiosis II occurs to this secondary oocyte. Purpose of meiosis II is to separate the sister chromatids. I get half of each of the chromosomes over here. There's my chromatids. Again, the other one goes into the other half, goes into the polar body, and it disintegrates. So this is my cell ready for fertilization.